Okay, so electrophysiological techniques are based upon the electrical activity of neurons. The fact that brain cells carry electricity along their lengths, they carry currents along their lengths, and so do some astrocytes as well. So those of us who've studied like sort of electrophysiology, like to say that we're actually recording the activity of brain cells. This is what the brain does. Some of these other imaging techniques we're going to see are based upon measures of changes in blood flow to specific areas of the brain, and that typically follows on. For example, an increase in activity. You can see an increased flow of blood into a region to support that enhanced activity. Remember that the astrocytes help to mediate that detection of enhanced synaptic signaling, greater release of neurotransmitter, and they couple that to expansion or dilation of capillaries in that region to bring in more blood flow. But electrophysiology techniques are not measuring blood flow like we'll see functional MRI, for example, or PET scanning. What they're doing instead is they're measuring the actual action potentials, the frequency of action potentials, the electrical activity that is brain activity. They're measuring brain activity. Wilder Penfield is a very famous neurologist who worked at the Montreal Neurological Institute in the 1950s, and he would often perform surgeries on those with intractable epilepsy. So epilepsy that did not seem to be treatable, at the time by drugs or other therapies. So he would actually, remove the skull and the piece of skull, and then the dura cut through, and get down to areas of the brain where the seizures were originating, and then he would test different areas, with an electrode, and he would often have a notebook there, and he it would be numbers. And he would say number one, and stimulate it. And if it, let's say, it interrupted speech, for example, maybe it was on the left side in the frontal lobe, and the person stopped talking in the midst of this, he would write down, it's interrupted speech expression or something along those lines, and he'd put the little number directly on that part of the patient's exposed brain, and then if he found an area that didn't seem to generate any kind of response, well, then he would that was a candidate potentially for removal, to help to prevent the onset of additional seizures post surgery. So one of the things that's interesting about that electrode stimulation, basically, of the cortex, is how for example, this is how we know pretty clearly. One of the reasons we learned that this sulcus right here, this like valley right here, right just centrally located right here. It's one that typically doesn't go all the way down. This usually like a little lip of gyrus here before it hits the lateral fissure, right? Here's the frontal lobe, right? Here's the parietal lobe. That central sulcus divides the frontal lobe from the parietal lobe. And, this gyrus that is, just before the central sulcus, this is called the precentral gyrus. Along that is a mapping, basically, of motor output to the opposite side, the contralateral side of the body. So if you stimulate, towards the very top, you get hip movement. If you are able to go into the longitudinal fissure, you could get, the feet to move. But as you go down, you'll hit the arm and the wrist and the hand, and then all of a sudden it flips to the face. And then the very lowest part along here is going to be sort of the tongue, the larynx, the pharynx. Basically, this is the homuncular mapping, right, of the opposite side of the body. And it's interesting because, it's not a one-to-one mapping. There's much more representation, right, in cortex for the hands and the face and the larynx and the pharynx. And interestingly, this is organized in ways that the temporal delivery of stimulation matters too. So if you do a brief pulse, right, you'll get like the hand to move, for example. But Georgopoulos is a very well-known neuroscientist at the University of Minnesota found that if you, basically stimulate it for like a half a second, like 500 milliseconds, you can get like a macaque's, hand to move forward and, come up to the mouth from whatever position it happened to be in. So there's an analogous, right, mapping of the contralateral side of the body. If you go, here's the precentral gyrus, here's that central sulcus, central valley, right, and you go post-central, right, over the parietal, post-central gyrus here. There's a similar homuncular mapping of the contralateral or opposite side of the body in terms of touch, right, in terms of touch and position. Again, with, exaggerated, areas for the hands and, the face and the larynx and the pharynx, and less for like, the back and the, behind the knee and things like that. And so the electrical nature of information flow in the brain, has been kind of utilized, using these electrophysiological techniques to explore, the relationship between, activity and function. And, there have been opportunities where, like I suggested, stimulation is delivered, to generate interruption of normal functioning, right? But there's also been significant recording from, the electrical activity of the cortex. There were many, many studies that were done early using electrophysiological techniques that involved inserting a single electrode and putting it right next to a single cell and recording activity, let's say, from a cell in a specific column of neocortex. So, the studies of Hubel and Wiesel, for example, on primary visual cortex, where they found that individual columns were responsive, functionally to bars of light oriented in particular, orientations, were done using this sort of single cell recording technique where they take an electrode and stick it down into one column of neocortex and record its response to the presentation of stimuli, optimal stimuli in different receptive fields. But now, more commonly, they often use what's called multicell recording. In fact, there's been a lot of kind of development, many advances made in the ability to implant arrays of electrodes over, for example, areas of the frontal lobe, which can then be record ongoing activity and then actually wirelessly send that to prosthetic devices. So you can bypass, for example, spinal transection if somebody's been paralyzed, by a spinal injury, for example. So multiple cell recording has become, very popular. There's a lot of interesting research on it, and it has a lot of, fascinating sort of clinical applications, therapeutic applications, potentially also commercial applications as well.